Good evening to all. I am requesting today's resource person, Professor Harinath Chakrapani sir, to unmute yourself. Sir, okay, thank you sir. We will start our session. Okay sir?
Sure.
Yes, Shubham Dapre sir, please start the session.
Good evening to all. It gives me immense pleasure to extend to you a very warm welcome on behalf of the Atlasa Prepitra College, Devruk, who has clubbed all of us for the 3 days national seminar on current trends in medicinal chemistry. I welcome our today's resource person, Professor Harinath Chakrapani sir, who is Professor and Dean International Relation and Outreach, ISER Pune. I welcome all the participants who have joined us on behalf of our college. Welcome everyone. Then I would like to request Professor Dr. Amit Varare sir for a welcome remark and introduction of our today's resource person, Professor Harinath Chakrapani sir. Over to you, Dr. Amit Varare sir.
Good evening to all. It gives me an immense pleasure to introduce today's resource person, Professor Harinath Chakrapani from ISER Pune. He has completed his BSc from Loyola College, University of Madras, and MSc from IIT Madras, and PhD from USA. He is CRSI Young Scientist Awardee and has been a DBT Innovative Young Biotechnology Fellow. Currently, he is working as Professor of Organic Chemistry and Dean for International Relations and Outreach Program at ISER Pune. He has 1,774 Google citations, 25 H index and 48 I-10 index to his credit. Under his able guidance, currently 10 PhD students, 1 postdoctoral fellow and 3 BSMS students are working at ISER Pune. He has published 58 research papers in various reputed journals like Journal of Medicinal Chemistry, Chemical Communications, Organic Letters, the Journal of Organic Chemistry. In addition to this, he has written various reviews and book chapters. He has delivered various talks at national and international level. Currently in the year 2021, he received Chemical Research Society of India Bronze Medal. He has received various research funding agencies from DBT and DST. Some of them are for the year 2017 to 2020. He received rupees 98 lakhs 46,000 from DBT, and for the year 2018 to 2020 he received rupees 81 lakhs 4,800 from DBT. With this brief introduction, I request you, sir, to start your talk on generating hydrogen sulfide to mitigate oxidative stress. Please, sir.
Yep, thank you, Amit, and thank you all for the invitation. It's a great pleasure to be here. And I got to know that some of you have done the NPTEL course in medicinal chemistry.
Yes, sir.
So I'm very delighted to hear that. Actually, this medicinal chemistry, I've been teaching for a few years, but then I thought, okay, let me just try and do an NPTEL course on this. And anyway, so I welcome your feedback. If there are anything that I need to do to improve the course, that will be good. Just to let me know. Yeah. Okay. So thank you once again for the opportunity to present my work. I'm just going to share my screen now. I hope all of you can see the screen.
Yes, sir, it is visible. No problem.
Okay.
Yes,
I'm going to turn off my video because it's a little just to conserve
okay, no issue, sir.
Okay. So okay. So the what I'm going to discuss with you is today is a project that I've been working on for 5-6 years now, which is basically working on trying to generate H2S, which is hydrogen sulfide. Am I audible?
Yes.
Yes, sir.
Okay, great.
Yes, sir.
And can you see my? Let me just make a pointer, laser pointer. Can you see the pointer?
Yeah.
Okay, great. Okay, so I think hydrogen sulfide needs no introduction. It's a very smelly gas that some of us have seen in our school labs. So this is the KIPS apparatus, which which I think, I don't know if it's still used in schools, but at least when I was in school, it was there, and it was a very distinct odor that we could see. And this is used to generate H2S. Okay. But H2S has a fairly long history. One of the earliest reports of H2S is in the 1700s, 1800, and so on, where in Italy actually people were characterizing this as an environmental toxin, and it was called as the sewer gas. And some of the symptoms of exposure to H2S include irritation and inflammation. Okay, so from that, so therefore this is the first report, at least that we are aware of, where it is characterized as an environmental toxin. And in the late 18th century, Carl Wilhelm Scheele discovered how to generate pure chemical H2S. And after that, there have been a number of sort of developments in this field. So in about 1803 and odd, this Francois Chaussier, what he did was he was looking at various effects of H2S on animals. So he used a horse model, and he characterized H2S gas as being quite toxic. And in late 19th century, that is in 1877, Professor Gaillon discovered or found that H2S is actually produced in bacteria. Subsequently, in the 20th century, there have been a lot of work done on H2S, and in 1942, the first report of H2S being produced in mammalian cells. So keep in mind now that we are talking about a sewer gas, that is something that is being produced in the drainage or the sewer, to now actually being produced in bacteria and also in mammalian cells. In the 1960s, there are a lot of papers from Russian labs where they show that H2S had very good effects, protective effects, especially helping in the cardiac and vascular function. So you're also seeing the change that H2S being a bad compound, a toxic molecule, to actually being something that is useful. So in the 1990s, 1996 to be precise, Hideo Kimura and co-workers reported that H2S is a neuromodulator. Since then, there's been a lot of work that's been done to study the effects of H2S as a neuromodulator, as a protective agent from inflammation, etc. So in a few years back, this compound ATB346 entered clinical trials for H2S. It's a H2S NSAID hybrid, and it entered clinical trials, and that's a very positive sign. So the question that we want to ask is, can H2S be used as a therapeutic? So as I mentioned earlier, there are a number of reports which talk about H2S being a neuromodulator, vasodilator, antioxidant, etc. But the problem is that you need to be able to generate it at a level where it is not toxic, because clearly H2S is toxic to us and to horses, as the previous report says. So one of the ways in which you can generate H2S is to actually use the inorganic salt. So sodium hydrogen sulfide is commercially available. You buy it, you put it in a pH 7.4 buffer, and what it's going to do is it's going to generate a burst of H2S. And there have been a lot of studies done with sodium hydrogen sulfide and Na2S, and it does show broad anti-inflammatory activity, and it mitigates or it inhibits the production of ROS, reactive oxygen species. I think this audience doesn't need an introduction to ROS, but these are toxic or oxygen-derived radicals which can damage a number of cellular components. But the problem with this is that once you make a solution of sodium sulfide, it just escapes from buffer, and you don't know how much actually that you are injecting into an animal model, or in case of tissue culture settings, you don't know how much is actually being added to cells. So therefore it's a problem, and we want to be able to improve this. So one way to do this is to actually think about a small molecule which on its own doesn't produce any H2S, but once it enters the cell, for example, it gets triggered or it gets activated, and then it produces H2S. So therefore there is no H2S production outside the cell, but once it responds to some stimulus, and this stimulus can be various things, it can be light, it can be an enzyme, or if you're looking at anti-inflammatory effects, it can be reactive oxygen species. But there are certain things that we need to understand. First of all, the compound must be cell permeable, and it should be non-toxic, of course, and the cleavage must occur in the response to the stimulus. So with this, we went ahead and looked at what are the ways in which one can produce H2S. So we came across this molecule called as carbonyl sulfide. So carbonyl sulfide, as you can see here, is basically carbon dioxide with one oxygen being replaced by sulfur. And as you know, carbon dioxide, there's an enzyme called as carbonic anhydrase, which cleaves off CO2 and produces water, and it produces carbonate and bicarbonate. So similarly, this carbonic anhydrase can also react with COS and produce H2S. And if you see here, the interesting thing here is that the oxidation state of sulfur here is -2, and the oxidation state of H2S, sulfur and H2S, is also -2. So there is no redox chemistry that is involved. So therefore this becomes a very attractive way to generate H2S. Now how do you, so now what we decided was we will sort of purpose this problem and make it into a compound that actually generates carbonyl sulfide. Now the way we designed this is Preeti, who just recently graduated from our lab with a PhD. She came up with this design. So we thought that COS could be produced from these thiocarbonates. So here, if you think about this, if X is nitrogen, then it becomes a thiocarbamate, and if you see the arrow pushing as shown here, then you can actually produce COS, and you will produce an amine. And now if X is oxygen, then a similar chemistry can happen, and you would produce an alcohol. Now how do you generate such intermediates such as 1? The way you would do this is, one of the ways you would do this is to look at intermediates such as this, where you have a quinone methite that's going to be formed through this 1,6 elimination, and this in turn can be masked as groups which can be cleaved under various conditions. So with this, we went ahead and synthesized these compounds. So we started with this picoyl oxymethyl chloride, converted it to the iodide, and then from the iodide we make this compound, which actually with this alcohol, with this benzyl alcohol here, which was then subsequently brominated with PBr3, and then you react it with thiourea and cleave the thiourea by thiosulfate to give you this thiol. And now this thiol can actually be reacted under a variety of conditions to give you both oxygen-based as well as nitrogen-based compounds. And the yields were pretty okay. Some of them were low, but we could repeat the synthesis enough number of times to get enough compound for our experiments. So with this, then we went ahead and looked at whether this compound produces H2S or not. So here, for example, when you add esterase, it cleaves this parallel group here, and it would get cleaved, produce formaldehyde and the intermediate that we're looking at. And one of the first assays that we did was to look at the formation of methylene blue. So methylene blue is a as the name suggests, is a blue complex, and it's produced when this amine is reacted with H2S in the presence of FeCl3. So what happens is that you get a very characteristic signal at 676 nanometers. And so when this compound was reacted with esterase in the presence of carbonic anhydrase, we get a nice signal, spectrophotometric signal, which corresponds to the formation of methylene blue. Now we also looked at the generation of H2S using an independent experiment. So here what happens is that HS minus attacks at this position, and there is an electron deficient carbon, and then it kicks out fluorescein. And so if H2S is produced, then you would see the formation of fluorescein. So indeed, when we expose MCF7 cells, pre-incubate MCF7 cells with this dye, and we add H2S, as you can see in the control, there is no signal, but and as we increase the dose of our compound, we see a signal in cells. So now what we mission of H2S want to do is to actually look at a stimulus which is ROS. As I mentioned earlier, oxidative stress is a condition that is associated with increased ROS levels, and these ROS are like, for example, superoxide radical anion, which can then get converted to hydrogen peroxide, and through Fenton chemistry it produces hydroxyl radical. Now hydroxyl radical is actually quite dangerous, and it sort of damages DNA and many other cellular components, and it causes stress. And elevated ROS is associated with many conditions, including neurodegenerative disorders. So we are very interested in trying to reduce ROS using this strategy. So one of the ways in which you can actually look for ROS, or one of the chemistry associated with ROS, is the reaction with H2O2. So here is a boronate ester which reacts with hydrogen peroxide. It's a very standard organic reaction, and it produces a phenol. So based on this chemistry, we thought that if you actually mask this compound as the boronate ester, it could get cleaved or oxidized and then produce this phenol, and the phenolate can then rearrange and give you the same intermediate that we were looking at earlier, which then should produce COS and H2S. So therefore, this strategy would allow us to specifically generate H2S under inflammatory conditions. So while we were doing this, we also decided that we will make a non-steroidal anti-inflammatory drug hybrid compound. So in this case, we made this mesylamine compound, and mesylamine is used as a NSAID in several clinical conditions. And in this project, Preeti was helped by Shweta, and Shweta is now presently a PhD student in Virginia Tech University, US. So we made this compound, and when we added H2O2, we find that we would expect that H2S would be produced. And so indeed, when we did this methylene blue assay, which I just explained a few minutes back, we do see a nice increase in hydrogen sulfide, which is the characteristic absorbance at 676 nanometers. And then we also did a number of experiments which I'm not showing in the interest of time, and we show that this was actually selectively activated by H2O2, and it's also well tolerated in cells, that is, it is not toxic to us. But the problem is, when we did the antioxidant activity, when we studied it, we find that it was good, but it was not very impressive. So we went back to the drawing board to see if we can improve this activity. And one of the ways in which H2S acts as an antioxidant is that it does what is known as a post-sulfidation to proteins. So what is this post-sulfidation? So for example, if you take a protein which has a thiol, under oxidative conditions, H2S can add another SH onto this. This is called as S-sulfhydration or protein post-sulfidation. So in this case, this is a protein complex which is a NRF2 keep 1 complex. NRF2 is nuclear factor erythroid 2 related factor, and Kelch-like ECH associated protein keep 1. So under oxidative conditions, once this gets post-sulfidated, what happens is that this complex breaks down, and NRF2 goes into the nucleus and binds to what is known as an antioxidant response element, ARE. And this allows for transcription of many genes, but some of them include antioxidant genes such as heme oxygenase 1, or a protein called as heme oxygenase 1, and glutathione S transferase, which helps in getting rid of electrophilic species, and NQO1, which is naphthoquinone oxidoreductase. So what we were interested in what we thought was that, is it possible to generate H2S under these conditions so that we can use the cellular antioxidant machinery to carry out the anti-inflammatory activities. So we decided to make this compound, which is a substrate for NQO1. And NQO1 is a 2-electron reductase. And so what we expect is that this quinone would undergo reduction. It produces this dialate. And if you see this chemistry here, this can do a ring closing, or a lactinization kind of chemistry, which then kicks out the same intermediate that we have encountered in the past, which should then produce COS and H2S. And again, we decided to make the mesalamine analog because it's used, it is used as an NSAID. So, I'm just going to show you some data that we have on this compound. And when we add this enzyme called as naphthaquinone oxidoreductase, we do see the production of H2S. And in this, I'm showing you the H2S electrode response data. So in the absence of the enzyme, there is no H2S produced, but in the presence of the enzyme and carbonic anhydrase, we do see a robust signal for the formation of H2S. And then what we did was we took colon carcinoma cells, DLT1, and when we take this compound, what we do is we expose these cells to oxidative stress, an ROS-induced compound. And so here is the viable cells under oxidative stress. And if you see protection, then this bar should be bigger. That means that the viable cells should be more. So when we do this experiment with this NSAID, we find that the NSAID alone is not very effective in protecting cells from ROS-induced toxicity, but our compound showed some moderate antioxidant activity. So if you see this difference, it's actually pretty, it is significant, but it is not very large. So for example, we're looking at somewhere between 20% and improvement to about 45%. So as a proof of concept, it's very, very useful, but what we would at best say is that it has moderate antioxidant activity. So to summarize this part, we designed these compounds that generate COS, and COS is the advantage of COS is that there is no redox chemistry involved to produce H2S, and we can install a variety of triggers. We've also done many other triggers to show in the interest of time, we can also produce an NSAID hybrid, but the antioxidant activity is not very impressive. Second problem is that we generate this electrophilic byproduct, which is called as a quinone methide, which is not very desirable. Of course, we are not the only people working on this. There are a couple of groups in the US who also work on generating these kinds of compounds. Now, when we look at it, we want to improve the antioxidant activity. So we moved to instead to generating persulfides. Okay, so let's compare the chemical properties of a thiol and persulfide. So here is a thiol, and typically the pK is around 7.6. But now, when you put a persulfide, the pK goes down, which means that this is actually going to be ionized to a greater extent. Not only that, the nucleophilicity is also much better. Okay, so therefore, persulfides have distinct chemistry compared to thiols. And another important thing about persulfidation is that when you take a protein under oxidative stress conditions, this sulfur can get oxidized to SO3 minus. And if this cysteine residue is catalytically important, once you oxidize this is an irreversible step, and therefore it's not going to be, that protein is not going to be functional. But now imagine that under these conditions that the protein actually gets persulfidated. That means you're adding an extra sulfur over here. Now under these conditions, if it gets oxidized, it produces SSO3 minus. And if SSO3 minus, if it's cleaved under conditions such as thioredoxin, then it produces bisulfite and it restores the protein. So therefore, the production of a protein persulfide is actually protective in nature. So being able to generate persulfides in cells has a very important effect in protecting cells from oxidative stress. Here is another experiment that shows that the importance of persulfides. So what they have done here is that they have looked at consumption of hydrogen peroxide. So H2O2, as I mentioned to you, is a reactive oxygen species, and under inflammatory conditions, the level of H2O2 goes up. So here what they have done is that they have generated persulfides under these conditions. So for example, here is GSSSG, and in the presence of glutathione reductase, it produces a glutathione persulfide. And I think many of you will know that glutathione is an important antioxidant. And then what they have done is they have compared the same under these conditions, the antioxidant activity of just GSSG, which will produce glutathione and glutathione, 2 moles of glutathione. So if you see here, the greater the percentage of H2O2 consumed, the better the antioxidant activity. So if you see glutathione, which is a very well-known antioxidant, is actually not very good at consuming H2O2. Whereas the persulfide is far better. Like if you see here, almost 100% of H2O2 has been consumed under these conditions, where only a few percent has been consumed by GSH. Not only that, this persulfide is also much better than H2S. So what we decided to do is, we decided that we should focus on persulfides if we want to improve the activity. So we have now redesigned the strategy, and what we are suggesting is that we will now work on a compound that generates, it should go inside the cell, it should get cleaved, and it should produce a persulfide instead of H2S. And what we wanted to do was, we wanted the stimulus to be again elevated ROS or H2O2 is what we are looking at. So now if you go back to your organic chemistry reactions, there is a reaction called as retro-Michael reaction. So the retro-Michael reaction is something like this. So you have an alpha beta unsaturated ketone or amide in this case, and the Michael reaction is addition of a thiol. The retro-Michael reaction is exactly the opposite reaction, where this kind of an intermediate is formed, and then if you push arrows, what is going to happen is it's going to regenerate the thiol and the alpha beta unsaturated amide or ketone, as the case may be. So based on this, what we proposed was that we could generate a persulfide through a retro-Michael reaction. So if you see here, the arrow pushing is pretty much similar to what we have in this retro-Michael reaction, and it would produce this alpha beta unsaturated aldehyde and a persulfide. Now if this is a benzene ring, then it produces cinnamaldehyde, and as all of us know, cinnamaldehyde is a very common constituent of cinnamon oil, which is approved by FDA as a safe compound, or it's recognized as a safe compound. So in order to generate this compound, we came up with this kind of a strategy. Prerna is the one who led this project. So here is a boronate ester. If you remember that this boronate ester can get cleaved by hydrogen peroxide. So it gets cleaved here. It would produce this enol, and this enol or enolate will then rearrange to give you the persulfide. So we made this compound, and when we add H2O2, we do see the production of loss of this compound 1, and we also did the kinetics, and we found that the rate constant was 0.0533 per minute. And when we look at the production of cinnamaldehyde, we get a rate constant of 0.1275, and it looks like these two values are the loss of this boronate ester is significantly slower when compared to the generation of cinnamaldehyde. And then what we do is, this persulfides are actually quite difficult to they are very short-lived species. So we trap this using FDNB, fluorodinitrobenzene. So if you see here, there's an electrophilic substitution reaction, and we get this product as here. And then using HPLC analysis, we measured the rate of formation of this adduct, and these two rates are actually quite comparable. That is, if you see here, this is around 0.15 and this is around 0.13. So therefore, what we suggest here is that the first step, which is the reaction with hydrogen peroxide, is the rate determining step. Then we went ahead and looked at the ability of this compound to protect against oxidative stress. Here we used menadione as a ROS generator, and if you see, menadione alone was actually was able to it's quite cytotoxic to cells, but then our compound was fairly good at increasing the number of viable cells. And here is the negative control. What we did here was this negative control actually gets cleaved and produces cinnamaldehyde, and so we wanted to check if the activity was due to cinnamaldehyde, and based on this experiment, we are confident that it's not due to cinnamaldehyde. So to summarize this part, we are able to produce persulfides, and we are able to get elevated ROS conditions to cleave it. But one of the problems that we are dealing with is the electrophilic byproduct phenolmaldehyde. And there are many groups across the world that are working on this persulfides. I've named a few of them here on top. And also, from India, Professor Pradeep Singh from IIT Kharagpur is also working on this persulfide donors. So we wanted to further improve the activity. So we went to look at the biosynthesis of hydrogen sulfide. So hydrogen sulfide is actually produced from cysteine, and I will not go through all the pathways here. But what is interesting is that when you take cysteine, and if you this is an enzyme known as cysteine aminotransferase, which produces mercaptopyruvate, and in the presence of alpha-ketoglutarate, it produces mercaptopyruvate and glutamate. Now, mercaptopyruvate is a very interesting molecule, and it can react with this, or it's a substrate for this enzyme known as mercaptopyruvate sulfur transferase, MST. And what it does is that it transfers this sulfur to MST and produces pyruvate. So we were very interested in this biosynthesis pathway. So I'm just going to explain this reaction in a little more detail. So here is the MST, which has a very reactive cysteine moiety. And once the cysteine moiety reacts with this mercaptopyruvate sulfur, you can see that the arrow pushing can happen in this following manner, and it produces pyruvate. This is the enolate of pyruvate, which can eventually produce pyruvate. And it produces this MST persulfide. Now, MST persulfide is known in the literature to further react and produce what is known as polysulfide. And polysulfides are excellent antioxidants. It can also get cleaved. The sulfur-sulfur bond can also get cleaved to produce H2S. And lastly, MST persulfide can also react with various proteins which has cysteine to produce persulfidated proteins. So keep in mind that we want to increase the number of persulfidated proteins because that helps with protecting the proteins under oxidative stress. So what we did was we went, we did some docking studies with the help of my colleague in ISO Pune, Dr. Arnab Mukherjee, and we found that the active site of this enzyme, this is the natural ligand that is docked here. Based on these studies, we came up with a few compounds which can sort of act as a substrate for MST. I'm not revealing the structure at this point because we have not published it as yet. But these compounds fit well in the active site, and we made a library of these compounds. The chemistry of these compounds is very simple. So we have a thiol over here, and this can react at this sulfur, undergo this kind of rearrangement as we see here, and it produces a ketone or a substituted ketone, and it produces MST persulfide. Now, when we do this, when we made these compounds and reacted with 3-MST, these artificial substrates were well accepted by this enzyme, and not just by human enzyme but also by E. coli enzyme isoform. And we see nicely H2S production under the conditions that we do the experiment. This work is done in collaboration with Dr. Amrita Hazra, who's also my colleague in ISOPOL. Then, as I mentioned, we are really interested in generating persulfide. So what we did was we took this electrophilic compound known as monobromobimane and reacted it with our substrate enzyme system in the presence of glutathione. So what we expect is that once glutathione gets persulfidated, then in the presence of this electrophile, it should produce this species, which is shown here, where n is equal to 1 and the mass is 530.138. So we did LCMS analysis, and we see a beautiful peak for the formation of this intermediate. And it also, this compound, this is produced only in the presence of the compound and esterase and 3-mercaptopyruvate sulfur transferase. So what I'm trying to tell you here is that this system that we have developed very nicely produces persulfides as well as H2S. With this result in hand, we went into some cellular studies. So what we did was we wanted to see whether this substrate was very selective to 3-MST, and so we, with collaborating with Dr. Deepak Saini Indian Institute of Science, Bangalore, his lab generated what is known as a 3-MST knockdown cell. So in this knockdown cell, what they have done is they have reduced the amount of MST present in cells. So what we would expect is that in cells which don't have much of MST, that our compound should not produce H2S. Here is an experiment where we look for H2S in cells. And what we also do is use what is known as a scrambled cell line, where they, this is like a wild type cell line. This has MST present. So here is the data for control. In the presence of a regular H2S donor, both cell lines where MST is less or MST is in the normal amount, you see a nice signal for this. But when we use our compound 1A, which is a substrate for MST, we see a signal only in scrambled cells. So these are the cells where MST is present, and here MST is not present, and therefore we don't see a signal here. So what this tells us is that this compound is quite selective towards the enzyme, which is 3-mercaptopyruvate sulfur transferase. This compound 6 is a negative control. What this means is that this doesn't have a sulfur, it only has oxygen, and so in neither cell line it's able to produce H2S. Then what we did was we wanted to look at whether this compound was able to quench hydrogen peroxide. And so here what we used was a donor that was generated in our lab, which we recently published. And this is actually a compound that generates ROS very nicely. So it gets cleaved by esterase, as shown here, and it produces this intermediate which actually reacts with oxygen and increases superoxide levels in cells. So this compound, this cellular experiment that I am showing here, is using a dye for H2O2. So when H2O2 is being produced, we would see a nice signal, a red color signal here. This is a near IR dye. And when H2O2 is not produced or basal level, you don't see much of a signal. Now what we did was we took these cells and pre-treated it with this persulfide generator. And what we would expect is that if it is good, then the signal should go down. And indeed, when we did the experiment, we find that the compound that we have designed is able to reduce H2O2 levels in cells. Then we went into an animal model. We first found out whether the compound is toxic or not, and we do see that and we were able to find that the compound is not toxic. And so we used an animal model for inflammation, where mice were treated with this compound known as lipopolysaccharide, LPS, and what it is it's a constituent of bacterial cell walls. So once the immune system sees comes in contact with this bacterial cell wall component, it increases inflammation, systemic inflammation increases quite a bit. So one of the markers of this inflammation is TNF alpha, which are these pro-inflammatory cytokines, and the other one is IL-6 in the brain. So the experiment we did here was we injected mice with the compound, then treated it with LPS, and then we did another injection of the compound. So in the untreated set of animals, you find here that there's a nice increase in cytokines, which is expected because of a broad inflammation. But when we treated with our compound, you see here this green color bar, we see that there is a significant decrease in the levels of pro-inflammatory cytokines in both TNF alpha as well as IL-6. Sodium hydrogen sulfide, on the other hand, did not have an effect under these conditions. So what this tells us is that our compound is able to reduce inflammation in the brain, which is really important because a number of neuroinflammatory disorders or neuroinflammation contributes to a number of disorders, including Alzheimer's and Parkinson's. Then we went to study another downstream effect of inflammation, which is the oxidation of prostaglandins. So oxidase prostaglandins. So as many of you know, this arachidonic acid gets converted to PGH2, which can get further oxidized to PGE2/D2 under oxidative stress conditions. Previous reports have shown that H2S can inhibit COX-1/COX-2 and reduce the levels of prostaglandins. So when we did this animal experiment looking at the oxidase prostaglandins in the brain, we find that again in the presence of LPS we do see a robust increase in oxidized, or in PgE2/D2 levels. This was done by LCMS. And when we treat it with this with our compound, we see a significant decrease in the levels of PgE2/D2. And again, sodium sulfide did not have any effect here, suggesting that our compound is far superior to NaSH. So these studies, we are wrapping up this project, and hopefully we will be sending out a manuscript very soon. But these studies tell us that we have now arrived at the right kind of compound, which can be used to reduce neuroinflammation, which was the target going in, which was to use H2S as an anti-inflammatory agent. And I think we are very close to achieving this goal. So to summarize, in order to understand, to sort of use the therapeutic potential of H2S, we need robust tools which can help generate and detect H2S. And also we should be able to dissipate or reduce the amount of H2S, because those have some applications in other areas. So our lab works on all of these three aspects, mainly on generation and dissipation. And we hope to continue to work on this in the near future. In addition to this, our lab also works on other gaseous molecules, such as nitric oxide and superoxide radical anion and peroxynitrate. So we work on synthesis of these compounds, and we also try to understand whether these compounds can be used as therapeutic agent. You will be all very interested to know that last week a nitric oxide generating compound was approved as a nasal agent against corona in Israel. It was approved a couple of weeks back. So with that, I want to thank all the people who are part of this group. This photograph was taken more than a year back. It is a pre-corona photograph. And we've had a number of people graduate from our labs, and I'm very grateful to all of them. And funding is from DST, DBT, and ISO Pune. And these are the collaborators that I work closely with. Generating this kind of data is not easy, and they have been very nice to me in helping me out in generating this data. And the last couple of slides on ISER. So ISERs, we have now ISERs all across India. And ours, Pune and Kolkata were the first ones, but now the latest one is Behrampur and Tirupati. So I don't know if there are plans to have more ISERs, but indeed I think we have made some little bit of a mark in Indian science. So hopefully we'll continue to do well. And the main program that we have in our in ISERs is this BSMS program, which is after 12th. But we also have a very strong PhD program, which is a 5-year program. And I welcome students from your college to apply to our doctoral program. So with that, I thank you all once again for the opportunity to present my work. And now I'll be very happy to answer questions that you might have. Thank you.
Thank you so much, sir, for your nice presentation. Now I am requesting audience to ask questions. Anyone is having questions? Sir, I think there are no questions
Okay, everybody understood everything. That's great.
from the audience side.
Okay.
Okay, thank you, sir, for your nice presentation, and I am very much thankful to you for accepting our invitation. Now I am requesting Ajit Devle sir to give vote of thanks.
Good evening all. Good evening all. On behalf of Good evening all. On behalf of my organizing team, it's my privilege to propose a vote of thanks on the second day of our 3 days national seminar on the current trends in medicinal chemistry. I would like to thank our today's resource person, Professor Harinath Chakrapani, for not only sharing his invaluable time for us, but also for the igniting us with his commendable talk on the subject. We all would see Sir expertise in his talk on the generating hydrogen sulfides in mitigate oxidation stress. He has also explained chemistry regarding to H2S, some reactions regarding to H2S, reactive oxygen species, then generation of persulfides, some biosynthesis of hydrogen sulfides, protections of neuroinflammations, very nicely in the liquid manner. I would like to thank our beloved principal Dr. Narendra Tendolkar sir for his guidance and support for this seminar. I also thank all the participants for patiently listening, and I would like to thank to head of department of chemistry in ASP College Devrut, as well as my colleagues and all faculty members in our college for their valuable contribution. Thank you so much.
Thank you once again for the invitation. I hope to have the opportunity to meet some of you at least in the near future. I wish you all the best for the seminar.
Thank you, sir.
For the invitation.
Thank you, sir.
काय सर मीटिंग आहे केली सर हे लाईव्हचं कुठली मीटिंग झालं ना बंद मीटिंग आहे फेल झाली त्याच्यात सेवा होत झालं ना मीटिंग आहे बंद केली झालं ना
आता जरा ते बघा क्लोज करा आणि युट्यूबला दिसते का बघा ती बाकीची पण असतील ना मुलं म्हणून
नाही
क्लोज करायचं
केलं की
नाही